be on the cusp of the next great era of wet AMD therapy. Which phase 3 data will guide physician decision making if new technologies are approved by regulatory bodies?
He's Greg Notstein, I'm Scott Criswanis, and this is New Retina Radio's ASRS coverage from Retina Today and Bryn Mawr Communications. Dr. Chirag Jhaveri spoke with us about anatomic data collected in the Phase 3 ARCHWAY study. How did patients who received the port delivery system differ from those on monthly Ranibizumab in terms of fluctuation in central subfield thickness?
And as FDA weighs the possible approval of faricimab for the treatment of wet AMD, we hear from Dr. Karl Csaky about the safety, efficacy, and durability of faricimab in the TENAYA and LUCERNE studies.
Those interviews coming up during this episode of New Retina Radio's ASRS 2021 coverage.
The literature tells us that fluctuations in retinal thickness among wet AMD patients might be linked with reduced visual outcomes compared to patients who have had stable retinas. Did patients who were treated with the port delivery system with ranibizumab experience fluctuations in retinal thickness? And if they did, was there any link to visual outcomes?
To answer this question, we turn to Dr. Chirag Jhaveri, who explored this topic in a presentation at this year's ASRS meeting. Dr. Jhaveri practices with Retina Consultants of Austin and the Austin Research Center for Retina. Dr. Jhaveri, thanks for coming on the show.
Thank you very much for having me, Greg and Scott.
Before we get into the PDS data itself, what can you tell me about the link between retinal thickness fluctuations and visual performance?
So recent data from Usha Chakravarti has showed us that fluctuations in anatomy can lead to worse visual outcomes. Post-hoc analysis from CAT, IVAN, Harbor, and the Hawk Harrier studies have showed us something similar. So we wanted to know if patients in the phase 3 ARCHWAY study who showed anatomic fluctuations and if that also affected their visual outcomes.
And why don't you give us that top-line refresher on ARCHWAY?
So the ARCHWAY study is a phase 3 study looking at the port delivery system, or the PDS system. And it's comparing patients who were given the PDS implant that allows a high concentration of ranibizumab to be delivered into the vitreal cavity, comparing that to monthly ranibizumab in patients who had wet macular degeneration. We found that the PDS system was equivalent to monthly ranibizumab with patients with wet AMD. The company has submitted this data in its filing with the FDA, and a decision is expected soon.
How did you determine whether a patient experienced fluctuating retinal thickness?
So what we did is that we assigned patients a fluctuation score. So this score was calculated over 10 monthly visits. And we measured the magnitude of change in their central subfield thickness, or CST. A change of less than 50 microns was considered negligible and was not included. So only patients who had 50 microns or more of change was that score, that amount added to their cumulative fluctuation score.
And a fluctuation score is your research team's own metric.
Yes, this is a novel method, and so it should be interpreted that way.
Understood. So tell us what you and your research team found.
So fortunately, only about 1 in 5 eyes in both arms, the PDS and monthly ranibizumab arm in the ARCHWAY study, experienced any fluctuations. Patients in the PDS arm who fluctuated had a mean change at week 40 compared to baseline of approximately 59 microns. And those in the monthly ranibizumab arm demonstrated approximately 19 microns in change from baseline. Patients in the ranibizumab arm had an average of 2.3 times or episodes of fluctuations during the trial. And the number of fluctuations in the PDS arm was an average of 1.8 fluctuations.
What did all of this mean for fluctuation scores?
So the mean scores in the PDS arm was 181, and in the monthly ranibizumab arm was 233. So the PDS arm had a total of less fluctuation compared to the monthly ranibizumab arm.
And how was that linked to visual outcomes, if at all?
So patients who had no CST fluctuations gained 1 letter in both the PDS and ranibizumab arms. However, patients who did have fluctuations lost 1.6 letters in the PDS arm and lost 1 letter in the monthly ranibizumab arm.
That's a difference of about 2 letters in all. Is that clinically relevant?
Yes, I absolutely think so. So we have to remember that this was at 10 months. We know that AMD is a chronic disease. So what we're looking forward to is longer term data out of the ARCHWAY patients, because we feel that it's possible that that difference between patients who had fluctuations versus did not have fluctuations could also continue to grow.
There is so much data in the Archway study. Tell our listeners what they need to take home from this talk.
So in general, in the Archway study, 80% of patients, regardless of the arms that they were in, did not experience clinically relevant fluctuations in CST, which was defined as more than 50 microns. Patients who fluctuated had similar visual outcomes in both groups, and which was less than patients who did not fluctuate. This echoes previous findings of fluctuations in fluid leading to worse visual outcomes.
Thanks for talking to us today, Dr. Jhaveri.
Thank you very much for having me.
A biologics license application was submitted to the US FDA in June for the treatment of wet AMD and DME. The drug, faricimab, and if it's approved, it would be the only bispecific antibody available for use in an ocular indication. The filing for wet AMD relied on data from the TENAYA and LUCERNE studies, a pair of phase 3 clinical trials.
We wanted to hear about the efficacy, safety, and durability of faricimab in TENAYA and LUCERNE, so we invited Dr. Karl Csaky to return to Retina Radio, where he will share details about his ASRS talk this year. Dr. Csaky practices at the Retina Foundation of the Southwest, which is in Dallas. Dr. Csaky, thanks for being here.
Well, thank you, Greg, and thanks, Scott, as well, for inviting me back.
Give us a quick summary of faricimab before we get started.
So faricimab is a very interesting molecule, and that is it's a bispecific molecule, which means that it has two portions of the molecule which bind both VEGF-A and Ang-2. And it really is born out of this idea that we know from our experience with anti-VEGFs that we are not really disease modifying. We have to continue to treat almost indefinitely these patients. And so the idea is that there are additional pathologic factors that are involved in the disease. Ang-2 has been shown in many animal models as well as in clinical data, is involved in an abnormal angiogenesis, which is the hallmark of neovascular AMD. And so as such, the idea of binding and inhibiting both VEGF-A and Ang-2 was a very attractive one scientifically.
And how were TENAYA and LUCERNE structured?
So the patients were randomized 1-to-1. In every trial, there is always a comparator, and in this case, it was a aflibercept that was given on label every 8 weeks after 3 monthly loading doses. However, in the faricimab arm, this was a very innovative design in which there was 4 monthly loading doses of the drug given, but then at weeks 20 and 24, disease activity was assessed. And depending on whether there was or was not disease activity, the interval was prolonged, which meant that patients had the opportunity to go beyond 8 weeks to 12 weeks, and then to go beyond 12 weeks to an every 16-week dosing schedule. So this was a very innovative way. It was also interesting in that it took the fact that there was an opportunity to predict at this early time point the disease activity interval, and that's what was done in this trial. And then the primary endpoint was primarily a change in best-corrected visual acuity from baseline over weeks 40, 44, and 48. And of course, this is an ongoing 2-year study, the results of which are still being assessed. But the good news was that from a baseline characteristics between all of the arms in both studies, they were well balanced.
Let's back up to the faricimab arms for a second. Why were all the patients in the faricimab arms considered to be in a single arm for the purposes of this study, rather than in 3 separate arms reflecting 3 separate regimens?
Well, the idea was no one knew for sure how many patients would end up in each one of these dosing interval arms, for example. So because of that, from a statistical and from a planning purposes, the idea was that all patients would be receiving faricimab, and then the dosing interval was going to be dependent on the patient's own unique requirement for drug. So in some ways, it was personalized, but the overall outcomes could be then compared in one group with 8-week dosing of aflibercept.
What did the researchers find at the primary endpoint?
So in TENAYA, the faricimab therapy resulted in a gain of 5.8 letters from baseline, while in that study, aflibercept gained about 5.1 letters. In LUCERNE, very similar, faricimab and aflibercept both resulted in a mean gain of about 6.6 letters. In short, despite these differences in dosing intervals, faricimab therapy was not inferior to aflibercept for visual function measures. In addition, using OCT, there was comparable similarities in central subfield thicknesses and the percentage of patients who avoided a major vision loss.
Let's go back to those faricimab treatment arms. Did anything break down as far as dosing schedule?
Yes. So as we said, this was a very innovative trial design in which at these early time points at 20 and 24 weeks, patients were categorized into a dosing interval. And at the end of the trial, at least in the first year, about 45% of patients were able to go to 16-week dosing interval. About 33% went to Q12-week dosing, and about 1/5 or 20% stayed on a Q8-week dosing schedule. What that means is that 80% of patients could remain on a Q12-week or higher or longer dosing interval than just 8-week. So in this sense, the median number of faricimab injections was 6 compared to the aflibercept arms, which was in a Q8-week dosing schedule and required a total of 8 injections.
Safety is paramount, especially when it comes to approval from FDA. Were there any safety concerns that were raised in the study?
Fortunately, there really wasn't. So the incidence of intraocular inflammation, which we know can occur with any drug we inject, was found to be very similar between both the faricimab arms and the aflibercept arm. So for example, with aflibercept, 1.2% of patients had some intraocular inflammation event, while about 2% in the faricimab arms. No difference statistically, but most importantly, as we've seen with other new drugs, there was no cases of vasculitis, occlusive vasculitis, or retinitis in these neovascular AMD studies.
All good information to know. Dr. Csaky, what's next for faricimab?
Well, as you mentioned in the opening, the FDA filing has been made seeking approval for patients with wet AMD. Of course, this study is ongoing, and I really think the 2-year data is going to be critical in our assessments of faricimab to improve the ability to increase the treatment intervals in patients. And of course, the study will go on beyond 2 years to a 4-year study as well, Avenil X study. So lots of additional data to come.
Well, we look forward to hearing more. Dr. Csaky, we will have to have you back on to give us more. Thank you so much for joining us today on New Retina Radio.
My pleasure. Be well.
That's it for this episode of New Retina Radio. Be sure to rate, subscribe, and spread the word about this podcast.
We've got 2 more episodes of ASRS 2021 coverage. Be sure to stick to your podcast feed to hear the updates.